let's quickly look at this IGCSE past paper 6 for February-March 2023, and this paper is for 1 hour, so I'll quickly try to run through it in the shortest possible time. So paper 6, alternative to practicals. First question is, alternative practical questions are usually 2 questions. You have question 1 and the second one. So I'm just going to have a quick run through of how to answer this question. Now, quickly, here is stated that potato cells take up methylene blue dye when the potato tissue is placed into a dye solution. Okay. Now, if the dyed potato cell are placed in water, the dye will diffuse into the water. So if the potato cell, the dyed potato cell, so you have to dye the cell in the potato before you place it inside water, and once that is done, the dye will diffuse into the water. So they said a student investigated the effect of temperature on the diffusion of this methylene blue dye from dyed potato cells. Also, the students use this method to do it. Step 1, they say you are provided with a beaker of cold water, a beaker of warm water, and a beaker of hot water. So you're provided with those 3, and those 3 will be more like our independent variable, because these are what we change in the course of the experiment. 2, let's say we measure the temperature of the water in the beaker of hot water. So that was done in step 2. Now in step 3, they say the label one test tube C, one test tube W, and another test tube H. Now draw a line that is 9 cm from the base of each test tube as shown in figure 1.1, and you place 3 test tubes in the test tube rack. So you have a test tube, you draw from the base 9 cm, you place them inside the test tube rack. And so in this case, we have 3 test tubes C, W, and H. So in step 5, they say they fill test tube C with cold water up to the line drawn in step 4. So the line drawn in step 4, which is 9 cm, you fill test tube C with cold water. Then you put test tube C into the beaker of cold water. Good. You fill test tube W with warm water, and you put into a beaker of warm water. You fill test tube H with hot water, and you also put into a beaker of hot water. Then once you've done that, so next, from this whole step we've been able to describe, we have not added the dyed potato cell. So here they said you are provided with 3 potato cylinders. Now they are being soaked in methylene blue solution and then rinsed. Soak in methylene blue solution and then you rinse it. Now place the 3 potato cylinder on the white tie and cut. You should cut the 3 potato cylinder to approximately 2 cm in length. So approximately. So this can be an error because the 3 potato cylinder or the wheel might be of different sizes. So so instead of measuring approximately trying to cut the length, maybe you should just measure the mass, so it will have equal mass. Okay, now step 9, they said place one of the potato cylinder from step 8 into each test tube. Then start a stop clock and leave the stop clock for 15 minutes. So that is a control variable. Now after 15 minutes, measure the temperature of the water in the beaker of hot water again. So here they said figure 1.2 shows part of the thermometer during step 2 and step 11. So what happened in step 2? In step 2, we measure the temperature of the water in the beaker, while in step 11, we measure the temperature again in the hot water. So step 2, what is the temperature at the start? So in between these 2, if you have 60 here, you have 65. So the difference is 5 degrees Celsius. So that means, and you have 10 smaller lines here. So for each 2 of this, it represents 1. So this will be 61. This will be 62. You have another 2. This will be 63. Then you have 64 before 65. So this value here should be around 61. The next one here, we have 35, and the difference between these two is the same 5 degrees Celsius. So each 2 will represent 1. So this will be 36, 36. You have 37, and the last one here we give you 38 degrees Celsius. So this will be 38. That's all. Then let's say remove test tube C, W, and H from the beaker and shake all the test tube for 10 seconds. So all the test tube were shaken for 10 seconds. That makes it a control variable also. Then you place the test tube into a test tube rack. Then observe the intensity of color in the liquid test tube C, W, and H. So the intensity of color in the liquid after you remove from the beaker. Okay, so the first question here is, figure 1.3 shows a student's notebook with their observation. So they want us to, first question here is prepare a table to record the result as shown. Okay, so this table is like one of the most simplest table I've ever seen in Igcse. So the first thing you need to do is, we have our set of values, which is this. So this is the test tube. So, and why the next one is the color. Why this value here has to do with the color, color of the liquid. This was color of the liquid after like 15 minutes. So what you draw, you just have a table that we have 2 different columns. You have test tube, and you have color of the liquid. So once you've done that, we have test tube C, W, and H. So CWH, and which is pale, medium, and dark in terms of color. Pale, medium. Okay, it will be W is medium and H is dark. So that's what it represents. So that's all for your 2 marks. Then the next question here is to state the function, state a conclusion for this result. So it's quite a simple conclusion. So you find out that the higher the temperature, the higher the rate of diffusion. The higher the temperature, because this is dark, because more blue dye diffuse out. So the higher the temperature, the higher the rate of diffusion. That's all. So the next question here is for us to, I think the next question is identify one possible source of error from step 8. I think I identified that while answering this question. Going back to step 8. Okay, the potatoes might be of different sizes. So you come back, the potatoes might be of different sizes. So that's all. The potatoes are approximately the same size. So instead of measuring the size, possibly you should measure the mass, and the mass should be equal. Okay, now they say identify one safety hazard in its investigation. So first you'll be working with hot water. So what you should do is make sure you use your gloves. Now, next is the reading on the thermometer in figure 1.2 show that the maintenance of temperature of the water during the investigation was a source of error. Yeah, so suggest an improvement to reduce this error. So the improvement you should look at is you should use, it was put inside a beaker, but you should use a thermostatically controlled water bath. Controlled water bath. Next, we're told here that the student did not repeat the investigation and only collected one set of results. Explain why it is important to repeat an investigation is to make it more reliable, also to calculate average, and also to which help to remove anomalies. You're good with that? Okay. The B part of this question is, a student investigated the effect of surface area on diffusion. They investigated the effect of surface area on diffusion. So the student used this method to do it. They cut four cubes from a potato. Now each cube could be of different sizes. That makes it an independent variable. They are of different sizes. Put the potato cube into a methylene blue solution for 24 hours. That will make it a control V. Now, after 24 hours, remove the potato cubes from the solution and rinse them in cold water. Now, fill 4 test tubes with water. Place 1 potato cube into each test tube. Leave the test tube for 15 minutes, another control variable. Now, some of the methylene blue dye will diffuse out of the potato cube into the water during the 15 minutes. Shine a light through the water in the test tube after 15 minutes. Now you should measure the percentage of light that is absorbed by the methylene blue dye in the water in each test tube. Now the higher the concentration of methylene blue dye in the water, the greater the percentage of light absorbed. So what was measured is this was being measured. So that would be our DV, what is measured dependent variable. Now the first question here is state the independent variable and the dependent variable in this investigation. And I think I've been able to identify them already during the course of reading. So I think the independent variable here should be the size of the potato cubes. So the size of the potato cubes. While the dependent variable is what we measure, which is here they look at the percentage of light that is absorbed by the methylene blue dye. So it's the percentage of light that is absorbed in the methylene blue dye. Okay, now state one variable that was kept constant. I think I identify a whole lot of them that were kept constant in the course of the experiment. So you just come, you write it. One thing that was kept constant is that the cubes were left for 15 minutes. That was constant in the experiment. So you find out that the methylene blue solution, the potato cubes were soaked in the methylene blue solution for 24 hours. And here, the test tube, you leave the test tube for the potato in the test tube for like 15 minutes. So any of those two will answer your question. Then the I part is figure 1.4 is a graph showing the student result. These are the student result of that investigation. Here we have percentage of light absorbed by the methylene blue solution. So this is usually your what is being measured. So this should be your dependent variable, and this is your independent variable. Surface area of the potato cube, size of potato cube, should be your independent variable, because your independent variable is always plotted on your x-axis, while your dependent variable is always on your y-axis. You need to take note of that. So even without reading this and from this graph, I should be able to get these answers right. Okay, the question here is, using figure 1.4, estimates the percentage of light absorbed by the methylene blue dye when the surface area of the potato cube is 18 degrees Celsius, 18 centimeters square. So show on figure 1.4, we should show it on figure 1.4 how we were able to do the estimate. So when surface area, so here I have surface area and I have 20 here, so 18 is in between, and I have 10 smaller lines and the difference is 5. So I know each 2 will represent 1. So this here will be 16, this next 2 will be 17, this here will be 18, and that is what I'm looking for. So I'm looking for 18. I'm going to trace this up to get the percentage. Okay, sorry. Okay, so this is what I need to get. Okay, so in between here, this is 60, this is 70, so the difference is 10. So I know there are 5 smaller boxes, so each will, if I divide by 5, I know each box will represent 2 units. So if this is 60, this will be 62, 64, 66. So my answer will be 66. So I've been able to identify this point where I take my reading from on the graph, 66. So that's all. Is solve this part plus my answer. That will give me my 2 complete marks. Now, question number 2, like I said, you just have 2 questions. So first, let's say a scientist investigated the nutritional content of fruit juice. State the name of the reagent or solution that will be used to test fruit juice for starch. To test fruit juice for starch, you need iodine solution. Now, the next question is, state the name of the reagent. State the name of the reagent that could be, or that will be used to test the fruit juice. Okay, sorry, I mean the second one. State the name of the reagent or solution that will be used to test the fruit juice for reducing sugar. So reducing sugar, you need Benedict solution. So a sample of fruit juice was tested for protein. States the positive test for protein. Usually purple color. A positive test will give you a purple color. It's just a recall question, so it doesn't give you a purple color. I give you a mauve color, whichever, you get your mark. Now, the next here is to plan an investigation, and we have a 6-mark question. So they said, plan an investigation to determine the effect of temperature. So we are looking at the effect of temperature on vitamin C concentration in fruit juice. So this is our, so when planning an investigation, let me just say certain things quickly to guide you. When planning an investigation, you need your independent variable. And from here, we know our independent variable is temperature. So we need to explain how we were able to alter the temperature in this particular experiment. So we need first, our independent variable is temperature. So, and approximately try to use at least 3 different temperature. 3 different temperature. It goes a long way. So 3 different variables that you're going to change, at least minimum. Then on vitamin C concentration, so if you look at your independent variable, another thing you should look at is things that will be kept constant. So since we are looking at vitamin C, I need the same fruit juice, the same juice. I need same volume of juice, same volume of juice. So you should be able to state it. Possibly you're using 3 cm cube of juice. It should be stated there. And what else can I keep constant? So the time also for the investigation. So look at your time, should be constant. So independent control. So you should look at your procedures, which you are going to describe when using these key terms or keywords in it. Then after that, you should look at your dependent variable, which is what is measured. What is measured. After what is measured, you should look at reliability. Now, your reliability doesn't change. You just write, you repeat the experiment, and you take average. Then you look at your safety. Now, so if you're able to get this is 6-mark question. So this is 1, 2, 3, 4, 5, 6. You'll be good. Simple as that. So you can take note of that. When writing the 6-mark question, you should make sure you have a touch of all these 6 key terms in it. So let's plan this investigation. Why am I even cleaning this? Okay. Now, the first thing is that I will do is to get 3 test tubes, which I'll label ABC. So I will say get 3 test tubes. Three test tube labeled A, B, and C, and I will add let me say 4 cm cube of fruit juice into each test tube. Then next, I will put test tube A into a controlled water bath, of let me say 25 degrees Celsius, and test tube B into 36 degrees Celsius, and test tube C into like 45 degrees Celsius. I'm gonna do that. So once that is done, next thing you need to do is after I will add like after, let me say sorry, let me bring in my time here, which is also a control. So I can say after 10 minutes I will remove each text tube, and add drops of DCPIP solution, which is used to test for vitamin C. DCPIP solution. That's it, and you record. So I will now record the time, the number of drops needed before it becomes colorless. Because a positive test for vitamin C, if you add drops of DCPIP solution, it becomes colorless, because DCPIP is a blue dye. It changes color to colorless if if vitamin C is present. Now with that, I think I've been able to cover the order, everything I need for my independent. I altered this temperature. Independent, I kept this constant. I kept my volume here constant. So the next thing is I will have to, and I also explain my dependent variable here, how I was able to measure the result. Then the last thing is to repeat. So I'll repeat. That's my reliability now. Repeat the experiment. And I can decide to say and calculate percentage change or average, whichever. Calculate percentage change in the number of drops. I can also come down and add, lastly, I'll add safety. I can see I wear protective clothing since I'm working with high temperature. So that's it. That will give you a full 6 marks. So the next part, which is part C of this C. Okay, let's look at part C now of question number 2. Scientists wanted to find out if drinking beetroot juice lower blood pressure. Now, 2 groups of men had their systolic blood pressure measured. Group 1 drank 500 cm cube of beetroot juice and group 2 drank 500 cm cube of apple juice. Now, after 6 hours, their systolic blood pressure were measured again. Now, the result is shown below. So we have group 1, group 2 at the start and after 6 hours. Now, the first question here, they say using the data, calculate the percentage change in the mean systolic blood pressure for group 1. So we're looking at group 1. So percentage change should be the new value, which is after 6 hours, minus the old value, divide by the old value, times 100. So the new will be 127.4 minus 132.4 divide by 132.4. If you just put this value in your calculator, your answer will become minus 3.8%. Now, this is state one factor that was kept constant in this investigation. So you go back, what was kept constant? If you look at this, they drank the same volume of juice. So the volume of juice was the same. You can just, the volume of juice, which is 500 cm cube. That's all. Now here we have the set of figure 2.1 is a photograph section through the bronchioles in a human lungs. Okay. Now they give us magnification here to be 130. So they said line PQ represent the width of the bronchiole. So okay, so this is line P to Q. So the first question is we should measure length of line PQ on figure 2.1. So length of line PQ, all you need to do is to just use your ruler. So I'm just going to give an estimated value here. So you place your ruler on this line, you measure it. Now where most students make mistake is check if your ruler is in millimeters or is in centimeters. Please, that's very important because your syllabus expects you to write down the answers in millimeters. So where most students make mistake is that we just write the answers once they measure in centimeters. So let's say your answer is 9.1 centimeters. All you need to do is from centimeter to millimeter, you multiply by 10. So it will give you like 91 millimeters for length of line PQ. Now they say we should calculate the actual width of the bronchiole. So we are calculating this. Now what was given to us also was the magnification. So using this formula, we know that our magnification is 130, so it will be 130 is equal to 91 over the actual width. So that, if you cross multiply over 1, you find out that your final answer will be 91 over 130. Sorry, I think this value here is 130, not 180. This is 130. So once you do that in your calculator, your answer will be 0.7 millimeters if you do that. So now they said make a large drawing of the layers of the tissue in the bronchioles. Now that means we should make a large drawing of this layers of these tissues in these bronchioles. You know, this is not like an art class, so you don't need to begin to shade or do any form of shading. You just need single line drawings, and your drawing should be very large to cover more than half the size or space given to you in the question. So if I meant to draw this, I will try and So I will have something. So you just need single lines, like if you will go back and check later. Single lines. You don't need to, but it should be quite really large. So we have some breakable lines that makes it round at the outside. Okay, so let me see if, okay, so there's a lot of space there, so which is similar. So I've been able to draw the outer layer. So the inside, what you should do also, you should try as much as to count how many is here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So you should try as much as you can to make sure 12 of those is able to enter into your outline. So if I just need to, I need 12 of those, you know, they're just, I know most of you can do better than me with this, so it's not, I have 1, 2, 3, 4, 5, 6, 7, 8. Okay, it's not up to that 12, so I don't really think I want to clean it. Okay, good. Let's have 1, 2, 3, 4. There is one that is okay. So I have 1, 2, 3, 4, 5. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now outer, I can decide, you know, they're just double lines, so you just okay, that's all. I think I tried. Yeah, that's all. That's what is required for you to do. So with that, you've gotten this, you get 4 marks. Now we have another question here, which is, they said a scientist investigated the effect of exercise on breathing rate and heart rate while running. The effect of exercise in breathing rate and heart rate while running. Now they measured the percentage increase in the breathing rate and heart rate from the resting rate during a 4-minute run. Now the results are shown in table 2.2. Table 2.2, I have the result here. Now let's see what question. They said we should, using all the data in table 2.2, plot a line graph on the grid of the percentage increase from resting rate against time. Now you will need to plot breathing rate and heart rate as 2 separate line graph. Okay, so because the graph is really down, I think I can just quickly do a graph here that I'll be able to see my data. So you can just convert it and put on your graph if you're practicing the same question. So quickly, we're drawing a line graph, and I told you earlier that this is usually your x-axis and this is your y-axis. S always comes before y, and so this is independent and this is your dependent variable. So once you identify that x always comes before y and you have more like a graph, you know, this is your x and this is your y. So what you should do first to get the first mark, everything that is here, you copy it down there to label your axis. So here is time. Everything, just copy it the same way it is per seconds, and everything that is here, you just percentage increase from resting rate. So percentage, please do not abbreviate, percentage increase from resting rate. Okay, my time is from 0 to 240, so I can just decide to start from just 10 in between. I can say 0, 60. After 60, I can add 60 again, 120. I can have plus 60, I can have 180. Plus 60, I can have 220. 60 plus 180, that's 240, and then I can have 300. So I think the largest value there is 240, so it's 60 difference between them. Then here, my lowest value and the highest value I'm looking at is 142. So I can decide to start with 0. I can decide to start with 20. If I have 20, I have 40. I have 50. I have 60. 70. Sorry, this is adding in tens. It should be in 20s. So 40. This will be 60, 80, 100. I think the largest value is 1. Okay, if I have 100, I have 120, 140, and lastly, I can have 150. So, 160. Once I have this, the next thing is to plot my data. So when it is 0, everything was 0, 60, 92. So when it's 60, I have 100, so here 90, so around 92. I'm just giving a rough sketch of how it will look like. When it is 120, it's 123. 120, I have 123. When it is 180, I have 185, 180, 140, 135 will be somewhere there. When it is 240, I have 142. So 240, I have 142. Then the same thing. So my graph for first one, I can just decide to use dotted lines to join them. So I can now have a key that will tell which each represents. Remember, I should be able to touch because it's a line graph. Then after that, I will do for the next heart rate. So 0, 0, 60, 21. When it was 60, it says 21. When it is 120, it is 40. Okay, 120, I got 40. When it is 180, I got 59. 180, 60, 59 before 60. Then when you have 240, it was 77. Okay, so 240, I have 77. So it's more like a straight line here. Join that. So once you do that, you can come up, you get a key just beneath your graph. You can, since we have two stuff, dotted lines represent breathing rate and straight lines heart rate. Heart rate. So that's all. You get the full mark. So you do that, you get 5 marks. Now state a conclusion. Conclusion from the data shown in your graph. You find out that breathing rate was higher from rest than heart rate. So if you look at this, breathing rate was higher than rest. So breathing rate is higher, percentage increase in breathing rate is higher from rest than heart rate. So breathing rate is higher from rest than heart rate. Okay, that's it. That's it on February-March 2023 Igcse paper. I pray this will be helpful for you in preparing for your exams.